morning, student, and welcome for today's lesson. I want us to understand a concept here that is based on inheritance, and we want to talk about the patterns of inheritance. Now we know that for a long period of time, for a century, people have been breeding animals and they have been growing crops. And in this, they have been trying to make sure that they get the more desirable character that they want from those particular crops and the animals. So this one has been based on them selecting this particular character that they want from the crops and from the animals. But they have not been understanding into details what is this particular character based on. So we have come up with an understanding that there is existence of the genes, so we go for this. These particular characters are controlled by specific genes. So that is what brings in the inheritance. So this particular character that an organism possesses can be passed to the offspring. And how is it passed to the offspring? It is passed to the offspring through genes inheritance. And that is what we want to base our argument on. So we want to understand the basis of inheritance. That is what we can now start talking about, the basis of the basis of inheritance. The basis of inheritance. So this is what I want us to understand. Now we know that the physical and the chemical characteristics that make up an organism is normally referred to as the phenotype. So there is this particular word here that I want us to understand, phenotype. This one we have said it is the physical or the chemical characteristics that describe an organism, what we are able to see from an organism, or what we can understand from that particular very organism is what we are calling the phenotype. So that is the first thing that we should be understanding. Now, the phenotype is actually as a result of the genes, the genotype of an organism. So we can say phenotype is the physical or chemical characteristics that describe the organism. So this particular phenotype, we have just indicated that the phenotype is partially the result of the genotype. So this is actually, it is coming as a result of the genotype. So there is this particular thing that we also have to understand, the genotype. So what is the genotype? Now genotype, we can define it as the genetic makeup of an organism. This particular genes that's set up to make up an individual organism is what we are referring to as the genotype. So we have got a genetic makeup of organism. So this one leaves us with the thought that characters of an organism are controlled by the genes of the organism. So that is what we are calling the genetic makeup of an organism, the genotype of an organism. That is what defines the character of an individual. So how is these particular characters really transferred from the organism? Now these characters are passed from parents to the offsprings. During the time that the offspring is being given rise to, these particular organisms will inherit these particular genes from the parents to the individual. Now, this particular phenotype or the character that we inherit, sometimes they can be influenced by the environment so that they don't appear like they were in the parents. That is what brings us to what we are calling variation. Like for example, we can have got a character that organism inherited from the parent, but that particular character does not show up like it was in the parent. This is due to certain environmental factors that can bring in variation. So variation is like the observable differences that we may notice in an organism based on what we understand from the previous organism. So that is called variation. Now, this particular feature or variation, they are brought up by a number of factors that are within the environment. So these factors that can bring in variation, they may come as a result of nutrition. Like we can talk about, we are saying variation. This is observable differences in organism. The observable difference in organism, and they have been brought about by the environmental factors. This include, like we can talk about causes of variation. Now, one thing that can lead to variation is nutrition. Nutrition. Now, what do you mean? Like, for example, we can see that if in case we grow crops, for example, we plant a pea plant, and then at one point we can talk about the pea plant producing the size of fruits in them. We can have large fruits, we can have got the small fruits. So if in case this particular pea plant inherited this particular gene for large fruits, let's say that there is large fruits here formation, that is the gene it was inherited from the parent. If this individual offspring is grown in an area that is less fertile, the fruits may not appear as large as they were in the parent plant, the initial plant. So it means that by that time, there is what is influencing the genes, the phenotype of this particular organism here is not going to appear like it should be, though the organism may have the genes. So by that, this particular factor here is influencing the appearance of that organism, the phenotype of that organism. So that is one thing that can bring in a change. Now, another thing that can bring in a change is when we talk about the environmental factors. Like if any case this particular pea plant was to grow well and produce for us large fruits, there must be those factors that help them make enough food. Like we have got water, we have got the sunlight intensity. So if any case those particular variables are not the way they are in the parent plant, the fruits that are going to be produced here, they are not going to appear as large as they were in the parent plant. So this one as a factor here can bring in this factor that we are calling variation. Now the next thing that can also bring in the variation is when genes are mutated. Like for example, you inherit a gene that is mutated. Now that one, if any case this particular organism is expressing a certain appearance in the parent, in the offspring it might not, because when the genes are already mutated, we have got a new meaning of those particular genes. So they are going to give us a varying character in the organism. So that is one thing that can lead to this particular variation. So that is the thing that I really want us to understand up to the point. So I want us to know the meaning of this particular term. These particular terms are very much important for us in understanding the genetics that we are going to talk about. So that is one thing that is very clear. So from here I want us to understand that why do we have got the differences in the genotypes between individuals? What are these particular factors that can lead to differences between different individuals? Let us try to see that. The factors that can lead to the differences in the genotype. Differences of genotypes between individuals of same species. Between individuals of same species. That is one thing that I want us to try to understand. So there are some two common factors here that can bring in the differences in the genotypes between individuals of the same species. Now I want us to look at something that we may be aware of or we have witnessed sometimes in the environment. We know that individuals can come from the same species, but they may not possess the same genes that they are supposed to be having. Why is that so? Like, for example, you may see that we have got sisters of the same father and the same mother. They don't have the exactly common features in them phenotypically. Why do we have this particular appearance in the organisms? Why is it so? So number one, we have to remember that there is rearrangement of genes during the process of meiosis. Rearrangement of genes during meiosis. This is one factor, rearrangement of genes during meiosis. Now, meiosis is a type of cell division that takes place in the reproductive cells. Therefore, it leads to formation of the gametes. And it is the gametes that will bring in the offspring. If any case during that particular time, every time there is that particular type of division, there is always genes being rearranged every time. So in the process, you may realize that a certain gene comes in a specific pattern that is not resembling the one that led to formation of a specific sperm cell or a specific ovum. Therefore, this particular ovum here, they might differ. If there are 3 of them, if there are 4 of them, all of them might be having different arrangement of genes pattern in them. And the moment the gene pattern is arranged differently, we will have different genotype of those particular individuals. Therefore, this one can bring in the difference between the organism of the same species. That is why you will see that brothers are not exactly the same. They are not like a clone, that is, because they may have the same genes that are arranged in different patterns. Therefore, they bring for us a different meaning that may not give the organisms to have the same thing. That is one thing that is affecting the organism. Now, we also have got the inheritance of the genes. Inheritance. We have got the inheritance of genes from 2 different individuals. Of genes from 2 different individuals in what we are calling sexual reproduction. Now we know that the diploid chromosome of a human being, for example, should be 46 chromosomes. We know that the diploid chromosomes from individual should be 46 in number. Now, this particular 46 chromosomes, we inherit them. And during this particular inheritance here, we inherit them through the sex cells. Now, each and every sex cells of an individual is a haploid cell. That is, it has got half the number of 46 chromosomes, 23. Now, during sexual reproduction, there is donation of these haploid cells. That is, the father will be giving us the 23 chromosomes, and the mother will also be giving us 23 chromosomes. So that is what is going to make up the complete body chromosome. That is the diploid cells that is going to give us 46. Now, if you look at it keenly, this particular inheritance is bringing in different cells which has got different genes together. Now, these ones here will be giving us different organisms with the different genetic makeup. Like you have got a half the number of genes from your father, you have got a half number of genes from your mother. So that one will lead to the variation. And then the next thing that you also have to understand that when these particular genes are being transferred, we have seen that the first one, there is rearrangement during meiosis. And during the passing up of this particular cells here, they are passed as a result of chance. There is no automatic pattern that will tell us that this cell will be automatically combining with this. It is just a matter of chance that one sperm cell out of millions can meet one ovum. So there is no specific order of out of these millions which one is going to fuse with the ovum. So that one will give us organisms that have got varying genes in them. So these two things here can lead to genetic genotypes that of individual of the same species not being the same. So that one can lead to the differences. So that is one thing that I want us to really understand very clearly. That is the first thing that we should understand. Now the next thing that I want us to understand is this particular gene pattern in an organism and how we inherit them from the female and the parent here. So the first thing that we have to understand is that we have got the pair of chromosomes in an individual, and we have said that we have got 23 pairs of chromosomes. So our body have got a complete set of chromosomes, that is 23 pairs that exist in us. Now this particular pair of the chromosomes, the way they are and the way they are passed, we have got those ones that are very identical. So they are the same. So if I have got a set of chromosomes that are the same, those particular similar set of chromosomes, we refer to them homologous chromosome. So we have got what is called homologous pair of chromosomes. Now homologous, in fact the word homo here means the same. So homologous pair of chromosomes are chromosomes that have got the same set of genes in them. That is what we are calling homologous. The chromosomes that have got the same set of genes in them, or the chromosomes that are the same with one another, that is called homologous. But is it true that the entire 23 pair of chromosomes are the same? The answer is no. The specific one that is different. Like for example, in a male human being, we have got the sex chromosome. The sex chromosome, for example. Now the sex chromosomes of the male, we have got the X and Y. So you realize that this is a pair of chromosomes. And then this pair of chromosomes that we have here, they are not identical. They are different. And that one means that even the sizes here, they are going to be different, and the genes that they are going to contain in them, they may have different number of the genes that are in them. So with that type of the chromosomes here, we call this particular chromosome as heterozygous. So the genes or the chromosomes in an individual can be of the same structure with the same genes in them, that is called homologous, or we can have those ones that have got the genes that are different in them, that is called heterozygous chromosomes. So in this particular manner, we can also try to understand what are the things that can come out of this particular individual, so that we can see the pattern of inheritance, how it is displayed in the organisms, the way it should be. So I want us to understand that one, that in every chromosome there are genes in them, and in this particular genes here, like for example, if I have got this chromosome here, this is a chromosome, I have got a chromosome here, this is the pair of chromosome, this is the pair of this chromosome. So those are pairs. So in this particular species, those species are called the locus, and in the locus we have got the genes, locus containing the genes. So there are genes in those particular chromosomes. Those spaces are not empty, they are filled with genes. So in the event that I have got a gene, for example, we can talk about a gene here, assuming that this gene is responsible for the height of an individual. So this person will be tall. That T, I want it to represent the character, which is the height of an individual, and the character specifically there is tall. So if any case this particular chromosome has got the the pair of it here, and they are the same. Now these are homologous pair of genes in a chromosome. They all stand for tallness. That is what we have. But the character tall here, the character height of an organism can express itself in different ways. We can have a tall person. We can also have got dwarf, the short person. So this is also a character. So you can find that in a certain chromosome, we have got the alternative form of that. Alternative form of this character here can be found in an organism, assuming that this is another pair of chromosomes. This is another pair that we have in this particular case. So if we have got a pair that in this case I have got this chromosome having a gene for tallness, and then in the other chromosome we have got the pair of chromosome. This chromosome pairs with this. So we have got this particular individual is having the gene for tallness, and in its pair we are having shortness. So this particular character here is an alternative form of this particular gene. Now this alternative form of genes, we are calling them allele. That is called allele. The alternative form of a gene in a pair of chromosomes, that is called an allele. So it occupies in the same position in the other chromosome. We have got the other character. So we have got two things here. We have got what we are calling homozygous chromosome, and then we have got the heterozygous, like in this case here, heterozygous. That is what we have. So homozygous, we can also have got another homozygous whereby an individual is having this particular chromosomes here. Like we can have this and this pair here. So we can have a T here and a T here. So looking at this, there are similar genes occupied in a locus, certain locus of the chromosome. Same to this one. So these are homozygous. Then this one is also heterozygous. That is what we have. So clearly, I want us to understand the meaning of the term heterozygous and the homozygous. So homozygous, we have agreed that these are chromosomes that have got the same pair of genes. Then heterozygous are chromosomes that do not have the same pair of genes. And in this case here, each one of them has got alternative form. So the alternative form of the gene is what we are calling the allele. Those are two different things. Heterozygous have said they are not having the same genes. And then this particular allele is the alternative form of that particular trait that we are talking about. Like the alternative form of tallness is shortness. That is what should be there. Now, in this particular individual that is heterozygous here, we may start asking ourselves, what will we be seeing in this person? What is the phenotype that we'll be seeing in this individual? Because we have got two different characters, two genes that are standing for alternative form in this individual. How will this particular individual look like? Now, what will determine in what we are able to see in a person is which of these particular two genes is dominant, is powerful, can express itself. So not all these genes will be having equal expression, or they are going to express themselves. There is one that will be expressing itself over the other. So the other one will be existing, but it is existing in a way that it cannot express itself. So there is a name we are referring to this particular gene. So the gene that can express itself over the other is called dominant. So there is this word here, dominant. The gene, this is the gene that can express itself fully. Express itself fully over the other. So if a gene can express itself fully over the other, that gene is going to be referred to as dominant gene. Now, assuming that in this case, for example, I have got this letter capital T to stand for tallness. Then letter small t to stand for shortness, or we can call it the dwarf individual. If we look at it, automatically this person here has got the two forms of the allele of the gene. This person will be tall. There is no problem with that. This person here has got two similar genes. This person will be short. There is no problem with that. But now the question comes in this individual that has got the two set of genes. What will be the character that we'll be seeing in this person here? Now this person cannot be tall, cannot be short at the same time. It has to be one. Now in this case, the gene that is going to, the phenotype that you're going to see, it means that the genes that stand for that phenotype is having the full expression over the other. That gene will be the dominant. In this case, assuming that this person is tall, we will therefore confidently say that this individual, this particular genotype here, this gene here, is having full expression over the gene for small t. Therefore, this person is having that character called tall. So in that case, again, this gene has been overshadowed. And this particular overshadowed gene is called recessive gene. The overshadowed gene is called, we can call it a recessive genotype. So this gene here is recessive. So it means that a recessive gene cannot express itself fully in a heterozygous state. It only has to express itself fully in the homozygous state. That is one thing that should be very clear with that. We should be clear with that. Now from here, I want us to look at the inheritance now. How can these particular characters now be passed to the children so that we can see how it works out for us in terms of the children. So we want to look at what we are calling the monohybrid inheritance. I want us to see what we are calling the monohybrid inheritance. That is one thing that I want us to understand here. So we can call it monohybrid or monogenic. We can call it monogenic, or we can be calling it monohybrid across. This is what I want us to try to understand in this particular chapters. So the first thing that we should know is that we have got what we are calling homozygote. Now we have got an individual that has got inherited the character from a parent. If any case we have got organisms that have inherited the same particular character from the parent, this one will be an homozygote. Like we have got one individual character, a child or an offspring, inheriting the same character from the father and also from the mother, so that we have got similar genes that are representing the same character that has been donated by both characters. That one becomes an homozygote individual. Now this particular individual will be referred to as a true breed. So we can be calling it a true breed, or we can call this condition true breeding. So this individual, we have said that it has got the same particular genes that have been, or character that are coming from the same parent. So it is going to have a same generation, a generation that have with the same character in them, based on what they are, based on the fact that they are inheriting the same character from the parent. So that is a true breed. It can easily be traced. So that is the thing. Like for example, let me assume that if there is that gene for shortness, the gene for shortness, for example, gene for shortness is inherited from father. The same gene is also inherited from mother. Same gene is inherited from mother. Is inherited from mother. That one means offspring will be gene from the mother and the father. That one now is referred to as monohybrid inheritance. So that is the case that we are talking about. Or assuming that we have got the similar gene, let us assume that I want us to use the plant that the first person that was studying the genetic use, we are calling it the pea plant. Pea plant. Now pea plant have got very much contrasting characteristics that make it so easy for us to understand the pattern of inheritance. So assuming that we have got two seeds, one that looks like this, we can call this wrinkled seed, and then we also have got another one that is like this. So this one we can call smooth coat seed. Smooth coat seed. Now assuming that this is a pure breeder, this is also a pure breeder, and then this is a condition, a character that can be passed through pollination. When we plant these particular plants together, they'll be crossing one another, they'll be crossbreeding, and therefore this one here is able to pass the genes to the offspring. This one can also pass the same gene to the offspring, but they have got contrasting characteristics here. One is wrinkled in the coat, another one is having a smooth coat. What will we see if any case they are crossbred together? Now if this is a pure breeder, they will not be having the same coat when we are having the offspring here. So it means that what will be seen will be based on which of this character is dominant. So assuming that the genes that is for dominant is coming from this plant, let us give it capital R, and then this one also has got a gene that is recessive. This is a smaller. So if this is a pure breeder, this is how the genotype will look like, and then this is how the genotype of this particular plant will look like. So if we plant them together, we will be seeing that at the end of the day, all the offsprings will be having seeds that are wrinkled. All offsprings will have wrinkled seed coats. And then somebody will ask themselves that what has happened to this other one here? Now, like we were saying before, in this case here, the genes are coming together, but one is having a full expression over the other. One is powerful, one is dominant. In this case, this is the one that is dominant. So it is going to overshadow these other genes that we are seeing here. So that is the thing that is going to happen for that individual. So we may not see the children having the same characteristics as the original parents here. They are going to be having different genes. If we continue with the pattern of spreading these particular genes together, you realize that only one character will be expressed. So this particular generation will be called first filial generation, which we can just refer to it as F1, first filial generation. All of them are going to have same characters based on the dominant gene carrying out the power. But if you carry out this generation and cross them again, you will now realize that there is that particular organism, the offsprings again for the F2 generation will be having some of them wrinkled seeds, some of them are going to have smooth seeds. Now, these particular wrinkled seeds and in the smooth seeds, they'll be coming out in a ratio. But that one does not mean that all of the wrinkled seeds are going to be purely wrinkled. They are going to have a mixture. Some will be purely wrinkled, some will be heterozygous instead. So there is that particular ratio of inheritance that we are having in this individual. Now we can express this one using a diagram expression that we can have in what we are calling the Punnett square. So assuming that we are crossing this, how will it look like? So let us see the pattern of inheritance here, how it's going to work out for us. So this particular thing can be expressed in a pattern. So we have got, I want to draw for you a Punnett square and see how we can inherit this. So number one thing, we have to know that we are having R capital to stand for, in this case, let us assume that they are having, we talked about it being wrinkled, So in our case here, this character become dominant, and then I have got a small one here being smooth. The smooth here I've said is recessive. So we know that it can only have a full expression if in this case it is in pure form. So we are talking about a trait whereby there are two true breeders that are coming in. So they are in their pure form. So we can say that in our crossing, I'll be having parents here, and I have got parent phenotype. So you know what a phenotype is, that particular outward appearance. So the first thing is that I'm having, the first phenotype is wrinkled seeds or coat. Then the other one is smooth seed coat. That is what I am having here. Then the next step that I'll be going to, I will be having parental genotype. I'm behind to have parental genotype. So how do I express these things in the terms of the genes? So genes here, I'll be having capital R, that's a pure breeder, and then small r, that is what I have. After this, I can have the parental gametes. So in the gametes, remember what we talked about, we are supposed to have the same genes, but we spread them out. I like cycle like this. They have to be a distance here to show that we are talking about the genotype. So that is what we have. So those are the genotypes that I have in those particular organisms. Now I want to cross. Now during the crossing of these particular individuals here, there is a chance of a certain gamete meeting the other. So I'll have my Punnett square here. This is my Punnett square. In my Punnett square here, this is what I have. So I'm having my gamete. So if I start with the wrinkled one, this is where my first gamete is. This is now where my second gamete is. Again, this is my gamete. This is my gamete. So this and this can cross, and I'll be having the genotype of the offspring here. I start writing the dominant one. That is how that genotype will appear. This and this, I will be having that one here. This and this, I'll be having this, and then this and this, I will be having this. So these ones are the genotypes of the offsprings. Now looking at it, if the capital one is dominant and wrinkled, it appears that all this generation will be having wrinkled seeds. So all the F1, F1 have wrinkled seeds. That is what is coming out. That is what we have. So that's the first thing that we should remember. After this, we can also talk about if we take the two of these organisms here and then we crossbreed them again. In this case, their state is heterozygous. So if we take the same two organisms here and then we crossbreed them, what will be the appearance? So our F2 here, F2 generation will come from, we'll be having again our new parents, parental genotype. How do they look? They are all wrinkled. We have talked about that. Wrinkled seeds. Then the other one is also wrinkled seeds. That is what we have. Their genotype Yeah, the first one was phenotype here, sorry. Phenotype. And then the genotype, parental genotype. We are having capital R, the small r. Gametes. Remember, these ones are now our new parents. So gametes will be, remember what we do to get the gametes? We have to separate them very well. So that is now what we have in the terms of the gametes. So assuming that we cross these two different individuals, what will be the outcome? So if they are crossed, this is what we expect. This is what we expect, that in the crossing, if this is our Punnett square, remember my gamete is this and this. So this and this I'll be having capital R, this and this, capital R and small r, capital R, small r, small r, small r. So in this we are seeing that we are having seeds that are wrinkled are 1, 2, 3. So 3 wrinkled seeds, and 1 smooth one. 1 smooth seed. That is now what we are seeing, that they are coming out in a ratio of 3 of them being wrinkled and then 1 of it being round seed. But those wrinkled one, they are not pure. They are mixed up. Some are going to be like they are wrinkled, some are not wrinkled. So that is the first thing that we are supposed to really understand here. So that is how we can really work out that and get out the differences of the two. That is what we can do here. The next thing that I want us to really understand is that this particular ratio can also be put in a genotypic ratio. So the genotype here can be in the ratio of 2 of them are we are having 2 of them are pure, or 1 of them. We can talk about if we start with this one, ratio that one to this one. So you can see that in the ratio, the genotype ratio is in 1 to 2 to 1, meaning 1 is homozygous wrinkled, heterozygous wrinkled, and then 1 smooth. So that is how we can get that particular ratio form coming out in this organism. But now the next question that we are going to ask ourselves, how do we know these ones here that they are heterozygous form and homozygous form? Now for us to come up with a way of knowing that whether the organism is in its pure phenotype form or in its heterozygous pure form, we have to carry out what's called a test cross. Now in a test cross here, you are using the unknown genotype of organism with a recessive known form so that you can really know what is found in the genes of those organisms. And this one will be expressed through the offsprings that will come on. So assuming that we want to carry out a test cross for this particular individual so that we determine. Now if any case we test cross them with pure breeding form, like assuming that we cross them with a pure breeding form here, the recessive one. In the event that no offsprings come out to be round seed, that one will mean that that individual was crossbred with the purely dominant gene, which of course is this one. So that one will tell us that this particular character is the one that we were testing for. In the event that we were crossbreeding them and then we find out that in the process some of them become wrinkled, some of them become round seeds, it means that the breeding that we were having there was not a pure one, the one that we are testing here. Therefore, it leads to the mixing up of the genes. So test crossing is basically to give us a clear view of what is the nature of the dominancy in the parent that we did not know is his gene. Is it pure one or is it a heterozygous one? That is how we do the test crossing, and that test crossing can help us to determine how the parents are. Like for example, assuming that this is what we do, assuming that we test cross and we know that if we test cross, this is what we should be experiencing. Assuming that I want to carry out a test cross so that I determine what is the breed of the organism that I'm having. So I have said that the first expectation is parental genotype. Parental phenotype here will be the one that I don't know is genotype is of course wrinkled. The seeds are wrinkled, and then the one I know is smooth. So I'm going to use a pure breeder here for the smooth one so that I can tell what is the nature here. So the parental genotype should be assuming that I have this with this, then parental gamete, I'll be having this, I am testing it out. So my Punnett square will be giving me this as the genotype. This and this, I'll be having this, this and this, I'll be having this, this and this, I'll be having this, and then this and this, I'll be having this. So in this case, all of these particular offsprings will be the same. So they are going to have wrinkled seeds. This one automatically will tell me, this one will automatically tell me that the genes here are the same. So I will know that the breeder that I was using here was a true breeder. So that wrinkled one, the dominant character that I will judge out of it is that it was having a pure form of that gene, the homozygous state. Suppose that I also cross and then find out this. We can also have another test here, parental phenotype. Parental phenotype. So you can see that the outcome here that is we are going to have is that we are going to see some individuals, plants here, having seeds with wrinkled coat. So we can have the ratio of them going to be at least wrinkled are in the same ratio as the smooth one. That is ratio 1 to 1. So this one tells us that the breeder that we are having in this case here was not a pure one. So in any case that we were not having a clear view of what was the genotype of the dominant plant or the organism here, the organisms that come out should be, if any case they are a mixture of our recessive gene also still coming out and the other dominants, we can now judge that that particular organism was not a true breeder. It was a heterozygous that we were having here. In the event that we were crossing them here, and then we get that all of them, our recessive character is now not being expressed. It means that they were both true breeders, and therefore the organism's characters that comes out here, they have been completely dominated. That is what we have there. So that is how we can carry out these particular genes and cross them. Otherwise, that is what I want you to understand up to this particular level. And maybe we'll be understanding how again we can be breeding the organisms. And now I want us to look at the codominance genes and what is it all about. Now, for us to understand this, we can take an organism like in the case of human being. So we can talk about in human, we have got, for example, this trait of the blood group, and how it is inherited. So we know that we have got the possible blood group here is ABO blood group. These are possible blood type that we have in human being. That one means that there is a antigen. Remember, blood groups is determined by the antigen. Then we have got a B antigen, and the absence of antigen, what we can refer to as O antigen. So these are the possible blood type that we have, or these are the blood type that we have. So in this particular case here, this particular blood protein that determine the blood type, the antigen A, the antigen B, and absence of the antigen will be giving us the different blood group that we have. Now this particular antigen can be coded and then being expressed in terms of their genes. Like we can have got a code for antigen A is this. Then we can code for antigen B in this way, and then no antigen can be coded for in that pattern. So this particular genes here can be given to an individual. They can be giving us the character here. Now these first two are always dominant in their own dominant, and then this one here is recessive, meaning it does not express itself fully when the two of them appear. So the possible genotypes of this particular individual, we can have the genotype of blood group can be expressed like this. Now when we have got this particular individual, we'll be having antigen A, and therefore this person is blood group A. Blood group A. Blood group A. Now when we have got this type of expression or type of genotype, this individual will be having antigen B, and therefore the blood type here, the blood group will be blood group B. And then when we have got this homozygous individual here, will be giving us no antigen, and this will be blood group O. That is what we have. That is what we have. So if this is the condition that we have here, we can also have this one here. This possibility is also here with us. Now in this case here, these particular two genes, they are all dominant. So they are all having equal expression. There is no one that is going to overpower the other. So this individual will be having both antigens. So we'll have antigen A and antigen B. So the blood type here becomes blood group AB. We will be having blood group AB. So this is the case that I want us to really focus our attention in. In this particular case, we are seeing clearly that this individual here is having both antigen because of the codes that we have. So the blood group becomes blood group AB. So it means that this particular gene for this code is not powerful and cannot express itself over the other one. At the same time, this particular code is not having a full expression over this. So their expression is a balance, a 50-50 expression. Now such type of expression, those genes are referred to as codominance genes. So codominance genes are two genes that have got equal expression without one clearly dominating the other. Such individual is referred to as codominance genes, and that is how the blood group is inherited. So blood group is one of the traits that have got codominance feature in them, just like we can have any other feature in different organisms that we'll be seeing. When the expression are equal, they'll be given the gene for both particular character existing in them. So that is one thing that we should be understanding. So the blood group here therefore can be inherited in this particular pattern, assuming that we have got individual of this particular nature. Suppose we have got these two human beings here marrying one another and having children. What can we be having here? Assuming that a woman of blood group O marries a man of blood group A. So assuming that we have got this parental phenotype, so if these are the phenotype that we have here, assuming that I have got the female here, that I'm using symbols to indicate who they are, this is the male, then parental genotype. Suppose I have got this particular genotype here, whereby this particular lady is having this particular set of genotype, and then this particular man is having this type of genotype here. What will be the type of the blood group that their children are likely to have? Gametes. So gametes here that we have here are going to be this one. Then if I bring in my Punnett square, so if this is my female human being, this is what we'll be having. Now this and this, I'll be having this particular genotype. This one I'll be having this. This one I'll be having this. So in this particular case, all the children will be having blood group A. That is what we'll be having. So that is a possible blood group inheritance that we can be having. Suppose we have got this individual here, we can be having another one, the parental phenotype. I'm going to have the female, the male and the female, then parental genotype. I'll be having suppose I have got this kind of individual, this one here, and I also have got this. What will be my possible blood group that the offspring will be having? So if we have got parental gamete, I'll be having this one. So my Punnett square will be giving me this type of result. So this is one of the gametes. So this first one will be having so this is the possible blood type that I'll be having. So in this case, I can have the individuals which have what all the blood groups available. Like this one will be blood group A, this will be blood group B, this is blood group A, this will be blood group O. So that is how we can get there to understand what is the meaning of the word codominance in an organism, and that is how we can inherit the blood groups of an individual. Now, moving on, I want us to understand that in some of these particular cases whereby we sample organisms so that we can get the clear values that we are coming up with, like we have got the theoretical values here. Like we are saying that if any case I have to express this, there is a possibility of the children having like all the blood groups here. If any case I talk about the organism like the one that we are looking at in the monohybrid inheritance, like whereby we talk about the ratio of the children coming out, there's a specific pattern that we are getting that is showing us how the children can inherit certain trait in the parent. But in a real life situation, this might not be true. Okay? This may not be true. These chances, the way they come, they may not be the same. So we may sit down and ask ourselves, if this is what we are getting, the expression in the genes, stamp of genes, and then an individual is having this particular blood type, like I'm blood group A, my wife is blood group B, then we are showing a case whereby our children should be having an expectation of having different blood type. But in the real sense, it might not be. We may feel that all our children have got one blood type. And then you start asking, what went wrong? What is the problem? Why is it not the thing that is the theoretical value that we are getting out of the crossing here on my board here is not the same thing that is on the ground? What can be? Now, in terms of sampling, there can be error. So we have got what's called the sampling error that normally don't give us the correct code. Like, for example, we have got there, like in terms of the children, we may talk about children. There is a chance of half of having different sex, like there is a half chance of the boy child being born, there is a half chance of the girl child being born. But in the real ground, you can find that somebody have got all the children becoming boys. What has happened to the other half of the girl child? So it means that in terms of sampling here, we can have what is called the sampling error. The sampling error. What is this error here, and how does it lead to us seeing that the actual samples here are not the same thing that we are seeing? So these errors, they come as a result of three possible things that can bring in the error. We have got the fact that we are reproducing, so reproduction here. Reproduction is not just coming in a specific pattern, but reproduction is a result of chance. What do we mean? We saw how the genes are arranging themselves. There is just a matter of a chance that at one point certain cell can meet the other one. It is not in automatic pattern that in this kind of reproduction, this X chromosome, this gamete with the X chromosome must meet with the ovum, or this the gamete with the Y chromosome must meet the ovum. It is a chance whereby any of this can happen. So this is one thing that can lead to an error. In the theoretical value, we are being shown that the chromosomes can meet in a specific pattern, like a Y chromosome can meet a gamete and an X chromosome can meet a gamete. That is what the theoretical value is showing us. But in the real thing on the ground is that it is a chance. In a matter of a million sperm, there is no specific chance that a specific one is going to meet the gamete. It is a matter of gambling with which one will be meeting based on the features that best fit it at that particular time. That is the first thing that we should remember. Number two thing that here will be giving us that the sample here may die. The sample may die. The sample of the offspring may die. Sample of offspring may die. So not all these particular offsprings that we have may be sampled and then they count here. We can have got a case whereby there is a embryo miscarriage. We can also have got you are planting the seeds, but these particular seeds here, they are not all germinating. So in that particular case, we talk about a sample is dying here. Therefore, it cannot give us a specific pattern that the theoretical value in the book was giving us. So in the ground here, the pattern may not be counted. So this one can lead to an error that may not give us a clear view of what the book is giving us. So that is one thing that can be here. Then we also have got insufficient sample. We may have inefficient samples. Like I may take this particular I may take this organism for study, like the dragonfly, for example. The dragonfly is what we normally use based on the features that they have. Now suppose I have sampled some of them. Maybe some of them can fly away, and therefore they are leaving me with a chance that it's not going to show me the real thing on the ground here. Yet the book is showing me that they are inherited in the same pattern. So this one shows that some of the errors that we may ignore during the sampling, but they are actually existing with us. Therefore, in the actual sense, they should not ignore them because they exist. But in terms of the theoretical value in the book, they assume that they don't exist. Therefore, they give us a specific ratio, like we have seen, ratio 1:1, ratio 1:2:1, ratio 3:1. So those ratios come because they are just theoretical value, assuming some of the happening. But in the actual life, there is a lot of the things that are happening that may lead to the organisms developing a specific pattern. So that is one thing that we should be understanding, and that is how the sample error is coming about. Then I want us to really understand the genetic. I want us to understand the genetic pedigree. The genetic pedigree diagram. The genetic pedigree diagram. So this is one thing I want us to understand. Now, we have been working out on the Punnett square that we have used to work out traits. So the Punnett square was giving us a specific theoretical value that is giving us a pattern of inheritance. Now, in the real sense, in this particular study of genetics, we may not be having a human being as our study. So we cannot use the human being as an experiment in most of the cases. This one is done by other animals. But we can be having some feature in the human being that we may observe, and we may use them to predict the pattern of that feature in a family. So for us to really understand this, we will be using a diagram so that we can just locate that particular feature in a human being, so that we can predict how the coming generation will be, so that maybe we may use that to avoid a situation, or we may use that to actually try to make sure that we are not in the disadvantaged form. Like, for example, there might be sex-linked traits that can be passed from one generation to another. And once we have studied this particular trait in different generations for a long period of time, we can now know that which particular individual has got this trait that may be affecting the family in terms of expression. So we can use this particular diagram to help us know this individual, and then we can advise, or we can help them to at least avoid the future occurrence of that particular condition. So that is why we can talk about the pedigree diagram. So I want us to indicate a pedigree diagram here and try to study how it works in this particular organism. So assuming that we want to look at the albino state of an animal here called the peacock, so I can use them like this. Let me have this diagram here. This was our starting point, and so this is parent number 1, this is number 2. Then from here I will be having this offspring. So this one here is with this one here. This is what I have. Let this one be number 3, number 4, and then I have this. That is what I have. This is what I have. So this is what this is my number 5 and 6. This one becomes my number 12 and 13. This is number 19, number 18. And then I will also have this one, number 10 and 11. So assuming that this one here is a diagram that is illustrating a feature character in a specific organism that I'm calling here peacock. So we are having, if I can explain in the diagram here, that if I see that, I'm talking about albino male. If I see this is albino female. Now this one is giving me normally pigmented male, and then when I see this is normally pigmented female. So these are some of the situations that we may be having in this particular organisms here. So so we have got this condition. So we have got an organism here that is showing two characters here. The organisms have got ability to have got the pigmented skin or the body color, and also can show the character of albino. So albino is a case whereby we have got the organism is not having the ability to synthesize the skin pigmentation color, hair, the eye. Those are different. So we have got like it's becoming purely white for that particular case. So we have got this particular individual here showing us a tree diagram of this particular family of this organism. And now this one is what we are calling pedigree diagram. So we can use this particular diagram to study and know at what point is which family affected, and in any case that we want to avoid the future occurrence of that, what is the precaution that that particular member of the family may take. So looking at it, look at our starting point. We have got our first two parents. We have got the male one here. We say that when you see that there is normal pigmented male. So this one here has got normal pigmentation. Then this one here is an albino female. Look at their children. Their children is number 4 and number 5. That is where their children are. So this child here is having normal pigmentation, and then this one here is also having normal pigmentation. That one is a clear indication that the gene for albino is recessive. It is not having a full expression. So this one here has dominated, so that we have got the children that look like they have got the pure skin pigmentation. And let us study and see what trends crop up. Now if you look at this particular individual ones here that we have, this one is having a mate here. which is number 3. This mate is not part of is not the children here. This is now a new mate coming in. But what are the children that they are getting? So these particular children, they are getting one with the normal pigmentation, and then the other one here is having the albino case. So let this one be my number 7, and number 8, and number 9. So we are seeing the scenario whereby out of this, there is at least one that is having the albino. So it means that this one could be traced back here. So this particular individual is a carrier here. This number 4, we can say that's a carrier. That is, and this one here may also be a carrier, because this albino here is expressing itself fully if we have got a case that is homozygous. So that is what we are getting here. So we are using this tree to track back where this particular individual is. So we can say that this individual is pure albino and got it from the carrier. So what advice can we give this if any case we want to stop the albino state in the future generation? This individual should have a mate that should be pure, normal pigmentation. Pure, normal pigmentation. That one can be helping this particular family to avoid that particular occurrence of the trait. What about this one? This one here may be a carrier, though he is having normal pigmentation. So the advice again that if we want to avoid this particular trait here, we must get the normal, he must mate with a normal pigmented organism, so that we avoid that particular scenario occurring in the future. The same feature can be studied here, and we can study the trait here, the albino here. This one here, we know how it might be looking like. So we have got albino here, meaning that this one was a carrier. So the two of them here, the gene become clearly expressed here. Now you can see in the family number 11, the generation that is giving us, so we may say that this one here is pure. Number 10 here is pure, because we are seeing their offspring here are having the normal skin pigmentation. So it's a pure for normal skin pigmentation. That's why it's dominating the albino trait that is coming from the parent number 11. Same to this particular side here. This particular individual is like, this one may be a carrier though, but normal pigmented. So this number 13, we may assume that this one is also a purely normal pigmented skin. That is why they are really suppressing that character, if any case we don't want. So the pedigree diagram is actually a family tree diagram that is helping us to track back a specific character that we may not want to have a continuity with. And therefore, it's really good for us to study there. So if any case these ones come in exams, we should be able to know how to track back using a family diagram. Then the last thing that I want us to remember, what we have been studying here, we have been studying some terms that have been very clear for us, that we have used examples here to try to understand and express this term, like the phenotype. Clearly what has come out is that the phenotype is actually the outward appearance of an organism. That is one thing that has been very clear with us. We have seen the word like heterozygous and homozygous pair of chromosomes. So homozygous was when the chromosomes are pure and they have the same genes in them, while heterozygous is a chromosome that has got the genes that are actually mixed up. There is no pure form, so we are calling them the hybrid. The other ones was called the pure breeders or the true breeders. We have studied the terms like the dominance, whereby you have seen with the expression that we have been having here, the dominance is a case whereby the gene is having a full expression over the other. So that gene is called a dominant, or it gives us the dominant character. And we also have with the recessive. The recessive was a trait that could not express itself fully over the other trait. So it means that it can only be expressed in the phenotypic form when they are in homozygous state. This particular character must be these terms must be well explained using the illustrations that we have given, so that we can be able to understand what's happening. Otherwise, I want to thank you for this particular time, and I want you to remember that you subscribe in our channel, and then we also advise you that if any case there is a question that you may ask from us, I will be ready to answer it on your behalf. Otherwise, thank you so much for it.